Constructing the CRISPR-Cas9 complex, also known as a ribonucleoprotein (RNP) complex, is a fundamental step in CRISPR-Cas9 gene editing. This complex is what enables precise targeting and cutting of DNA. Here's a breakdown of the key steps involved. 1. Design of the guide, RNA, gRNA. Identify the target DNA sequence. The first crucial step is to determine the exact genomic sequence you want to modify. Locate the protospacer adjacent motif, PAM. Cas9 requires a specific PAM sequence, typically 5-NGG3 for Streptococcus pyogenes, Cas9 or Specias9, immediately downstream of your desired target sequence. The Cas9 enzyme will only cut if this PAM is present. Design the 20-nucleotide spacer. This is the critical part of the gRNA that will be complementary to your target DNA sequence, the protospacer, located immediately upstream of the PAM. It's essential for high specificity. Choose gRNA format, single guide, RNA, sgRNA. This is the most common and convenient format, combining the CRISPR RNA and the transactivating CRISPR RNA into a single, synthetic RNA molecule. Two-part guide, RNA system, involves separate crRNA and tracer RNA molecules that self-assemble. While effective, it can be less efficient than sgRNA. Utilize design tools. Many online bioinformatics tools are available to help design gRNAs with high predicted activity and minimal off-target effects. Consider modifications. Chemical modifications to the gRNA can enhance stability, reduce immune responses in cells, and improve overall editing efficiency. Generate the gRNA. The gRNA can be produced through chemical synthesis, ordering pre-designed and chemically synthesized sgRNAs or crRNA, track rRNA duplexes. In vitro transcription, synthesizing gRNA from a DNA template using an RNA polymerase in a test tube. Plasmid expression, cloning the gRNA sequence into a plasmid that can be introduced into cells. 2. Obtaining the Cas9, nucleus, recombinant Cas9, protein. The most common approach for RNP complex formation is to use purified recombinant Cas9 protein. This protein can be obtained commercially or expressed and purified in-house, for example from E. coli. Cas9 encoding plasmids or mRNA. For some applications, Cas9 can be delivered as DNA in a plasmid or mRNA, which the cell then transcribes and translates into the Cas9 protein. However, using preformed RNP complexes is generally faster and can reduce off-target effects because the Cas9 and gRNA are only active for a limited. 3. Assembling the CRISPR-Cas9 ribonucleoprotein, RNP complex, in vitro. This is the core step where the Cas9 protein and gRNA come together to form the active complex. Combine Cas9 and gRNA. Typically, Cas9 protein and gRNA are mixed in a defined molar ratio. For example 1:1 or 1:1.2 Cas9 to gRNA, in a suitable buffer, for example nucleus-free buffer PBS. Example RNP formation protocol: dilute Cas9 to a working concentration, add gRNA, for example 2 mm stock, and Cas9, for example 1 mm stock, to achieve the desired molar ratio and final concentrations. Add nucleus-free water to the desired final volume. Incubation. Gently mix the components and incubate at room temperature for a specified period, for example 5-20 minutes. This allows the Cas9 protein to bind to the gRNA, forming the stable RNP complex. Optional, in vitro cleavage assay, pre-validation. Before introducing the RNP into cells, you can perform an in vitro cleavage assay to confirm that your assembled complex is active and can cut the target DNA. This involves adding a DNA substrate containing your target sequence and PAM, incubating with the RNP, and then analyzing the DNA for cleavage, for example via gel electrophoresis. 4. Delivery of the CRISPR-Cas9 complex into cells. Once the RNP complex is formed, it needs to be delivered into the target cells. Common methods include electroporation, nucleofection. These physical methods use electrical pulses to create temporary pores in the cell membrane, allowing the RNP to enter the cytoplasm and then the nucleus. This is a highly efficient method for RNP delivery. Lipid-mediated transfection. The RNP complex can be complexed with lipid-based reagents that facilitate its entry into cells through endocytosis. Microinjection, directly injecting the RNP complex into individual cells, often used for zygotes or difficult-to-transfect cells. Important considerations: nucleus-free conditions. It is crucial to work in a nucleus-free environment to prevent degradation of the RNA components. Optimization. The optimal concentrations, ratios, and incubation times for RNP formation and delivery can vary depending on the specific Cas9 variant, gRNA, cell type, and experimental goals. Off-target effects. Carefully designed gRNAs and optimized delivery methods help minimize off-target cleavage events, where Cas9 cuts at unintended sites in the genome. DNA repair pathways. After Cas9 introduces a double-strand break, the cell's endogenous DNA repair mechanisms, non-homologous end joining (NHEJ) or homology-directed repair (HDR), will repair the break. Understanding these pathways is crucial for achieving desired gene edits, knockout,